Hello my dear students. So in the first chapter, we discussed what classification is, and then we looked at the two main types of classification, artificial classification and natural classification. So there I discussed the nature of artificial classification. We looked at examples and we were able to understand, I'm sure you all were able to understand the drawbacks in artificial classification. Because there we consider only one criteria. So the relationship among organisms, especially the evolutionary relationships among organisms is not considered. Because of that, sometimes organisms that need to belong to totally two different groups come together. So that is why there was a need for natural classification. So now we'll try to understand what that is. Natural classification. A natural classification depicts the evolutionary relationships among living organisms. So when we say evolutionary relationships, you all know vertebrates are a certain group, invertebrates are a certain group. Likewise, algae, although they have green pigments, chlorophyll, they belong to a certain group, whereas the plants that do photosynthesis, they belong to a different group. So that is where the evolutionary relationships are important. So all that will be considered and it will be explained in natural classification. In natural classification, morphological, physiological, cytological and molecular biological features of organisms are considered. Now what are these? What do you understand by morphological? Morphology means the external features. Now we said no in the previous chapter. When you look at a lion, you can identify it's a lion based on its external characteristics. Now a zebra is different. A fish is different. Plants, a coconut tree is different from a mango tree. So all those are possible from the external features. So when we say morphological, those are the characteristics. Even in your class, you have about 30 students in your class, 30 or 40 students in your class. You can identify each person. We call them by name, but how do you identify the person? From their features. So that is what we consider under morphological characteristics. Then what is physiology? Now when we say physiology, we have the body of the living organism. Now inside our body there is the digestion process, respiration process, like that there are many processes taking place. But in addition to that, there is the cellular respiration, the synthesis of protein. Like that, there are physiological functions taking place within our body. The tissues, inside the cells, all the functions that take place even inside the body of both plants and animals, all those together is known as physiological functions. Then cytological. What is cytology? We have learnt about cells. Now you all know plant cells are different from animal cells. Then you all know viruses don't have a cellular organization. They do have living characteristics and they are non-living as well. So they don't have an organization. But if you take the bacteria cell, that's different from the plant cell and animal cell. So when we consider all these cells, their structure, what is present in their cell wall or whether cell wall is present or not, all the structures and the functions of all these components of a cell is studied under cytology. And then if we say molecular biology, related to molecules. For example, you are familiar with biomolecules. Under that we have studied about nucleic acids, DNA, RNA, how they are synthesized, how that synthesis is important for the organisms. All those are considered under molecular biology. So when we classify using natural classification, we consider all these characteristics. Morphological characteristics, physiological characteristics, cytological characteristics and molecular biological characteristics or features. So when you consider all that, you basically look at all the characteristics of an organism. It can be a unicellular organism, plant or an animal, but you consider all the characteristics. So the natural classification possesses below features. Because we consider all these, this natural classification possesses two main features. That is what we are going to discuss now. So if we consider those two features, I will write them first. Explains the natural relationships among organisms of the same species. And also it explains evolutionary relationships among organisms. So in this, if we look at these two features, it explains the natural relationships among organisms of the same species. Now, when you say species, those are the organisms that can interbreed and produce fertile offspring. So, those organisms are grouped as one species. So, when there are species, there are organisms. Under species, there are subspecies also. So, these organisms, for example, if you take a dog, there are different breeds of dogs. So, they might come under the same species or they might be different species. But if they are under the same species and they have different types, then you can understand the relationship between them. Then if we take the evolutionary relationships among organisms, we have been discussing this before also. When you look at different groups, but with similar characteristics. For example, wings or organs used for locomotion, appendages used for locomotion. So, the legs is one type of appendage, whereas the wings are a different type of appendages. Fins are different types of appendages. So, all these developed along the evolutionary process. So, that gives rise to or that explains the evolutionary relationship. So, as an example, if we consider the locomotive appendages. Example, we can say consider the appendages used for locomotion. Say there man uses legs. Then birds use wings. Then fishes use fins. So, these are different appendages used for locomotion. You can see fishes belong to one group. Then after fishes, we have the development of amphibia. There they have the legs, but you all can remember the legs of frogs. There is membrane between them. Webbed feet we call it. So, they can use that for swimming as well as hopping on the terrestrial environment. So, that is a development. From them, we have the reptiles. You all can remember reptiles, they walk on land, but they have feet which are short feet usually. You can remember the feet of crocodiles, lizards. So they do have feet but short. So that's a development. Then from there, if we look at the mammals, normally mammals have long feet, so they are usually above from the, they are at a higher level from the ground level. That's because of the structure of their feet or the appendage used for locomotion. Then if we take man, us, we stand on our feet. That's again the evolutionary development. So all these can be explained in a natural classification. So in this, you all can see these three are different groups. Man belongs to one group, birds belong to another group, fishes belong to another group. But there is evolutionary relationship. We all come under vertebrates. So again, some similarities there based on evolution. At the same time, if you take the organisms of a certain group. Now, if we take the mammals, we come under mammals. There are other mammals as well. We have the deer, cow, lion, all those, even dolphins, whales, bats, all of them come under mammals. But their appendages used for locomotion, it can be legs, but still the structure of the legs are different, as well as there is relationship. The bats, it's actually the limbs that becomes the wings. And in dolphins and whales, the limbs that become the flippers. So there also you can understand the relationship between the species of a certain group. So all that will be considered in a natural classification. I'm sure you all can understand that basic concept. So with that, I will move on to the next slide where I will just go through the differences between natural and artificial classifications. So here I have made it into a table so that you all can have an understanding. You will be able to compare the two types of classification. So we have the artificial classification and the natural classification there. So if we consider the properties used for classification, normally for artificial classification, we use presence or absence of locomotive appendages of organisms, or we use habitats of organisms. So those are the common properties used. Whereas in natural classification, we consider all these: morphological, physiological, cytological and microbiological features. I have explained what they are to you. Then if you look at the evolutionary relationships, the artificial classification does not explain the evolutionary relationship. Whereas the natural classification, it explains the evolutionary relationships among different organisms. That is one importance of natural classification, or why we really needed the natural classification. The same way, if you take the natural relationships that are again not explained in artificial classification. It does not explain the natural relationships, but in natural classification, it explains the natural relationships among organisms of the same species. So that is what we discussed in the previous slide. These 2 points. The features of natural classification. You all have to remember that, students. Then if we look at some examples for artificial and natural classifications, under artificial classification, we have used a few criteria before, classifying organisms as animals and plants. Then we consider the classification where we consider the presence and absence of legs, then the presence of absence of wings. Like that, we can use different criteria. I have given a few examples here. Number of stamens in plants. If you take different types of plants, you all know what stamens are. It is the male reproductive part of a flower. So the number of stamens can be considered as a criteria to classify flowers. But that again comes under artificial classification. Then presence or absence of wings. Then ornamental, herbal and poisonous plants. We have discussed all these examples. So there are 3 examples given to you. Like this, you can consider many more criteria. We have discussed that also in the previous chapter. But if you take the natural classification, we will need to know about 2 classifications. The 5 kingdom classification, I will just introduce that concept to you all. We will not be discussing that in detail, but the 3 domain classification, that is the classification that we use currently. The scientific natural classification that is used now. So we will be going into detail about the 3 domain classification. So those 2 are natural classification methods. With that introduction, students, I will move on to the next slide. So in natural classification, organisms are grouped into taxonomic levels in a hierarchy of categories. So here you need to remember or understand these 2 terms, taxonomical levels and hierarchy of categories. Now we said there are about 8.7 million species. Now to classify them, you can't just put them into different groups in one level. That's going to be difficult because as you can understand, now again and again I am telling you all this example. Now when we take mammals, we are mammals, there are so many animals, terrestrial animals, bats are mammals and whales are mammals. So especially when you consider bats and whales, they are very much different from most of the other mammals. But we classify them into mammalia because of certain features. But before we put them there, we have to classify them based on characteristics in different levels. First we consider them as animals, then under that what type of animals, like that we need to classify them. Same goes for plants. Now if you take coconut tree and a mango tree, you all know coconut tree is a monocot plant, mango tree is a dicot plant. But before putting them into 2 different groups and further dividing them into groups, we need to first understand that they come under plants. Then under plants also, they have a certain level of classification. Finally only they come to that simple group. So when we have that order, the hierarchy, starting from the highest group, which will include a very large number of organisms. But as you go down the hierarchical levels, you can come to the lowest level where there will be comparatively less number of organisms. So those are the levels that are used for classification. So under that, we call those levels as taxonomic levels. So taxonomy, naming. We can name these levels. Those are the names that we are going to consider here. So before I go to the extra knowledge part, you need to know all these taxonomical levels. We start with the domain. That is the topmost level. So in the previous slide, I told you all 3 domain classification. So all the organisms that are there on earth have been divided into 3 groups. Those are called the domains. 3 domain classification. So under domains, under each domain, we can have either one kingdom or more than one kingdoms. So the second level is the kingdom. So we have domain, then we have kingdom. Under kingdoms, we have phylum. That is the next level. Phylum. After phylum, here also the classification is given. We have the class. Then thereafter, we have the level called order. So domain, kingdom, phylum, class, order. If we take the domain, there is a domain called eukarya. Eukarya means the cell has organized nucleus. Here also I will explain that. So those organisms. So basically almost all the plants, multicellular plants and animals, all the developed plants and animals come under one domain, domain eukarya. Under that we have the kingdom Animalia. So there you know the animals go into kingdom Animalia, whereas plants go into kingdom Plantae. So two different groups. There are two more kingdoms as well. We'll discuss those. So there you can see the number of organisms in each kingdom decreases compared to the domain. Then from there we go to phylum. Under Animalia, you all know the vertebrates, invertebrates. Under invertebrates, again we have the different phyla. So there you all know about Annelida, Arthropoda, Mollusca, like that. There are different phyla. So there it is again, you can see the decrease in number of or types of organisms under each group. Then from phylum, we go to class. Classes under each phyla. Again, you're selecting some organisms and putting them into classes. Thereafter, order. After order, we have the family. Family, you're familiar with the Leguminosae family. When we discussed nitrogen fixation in your previous grade, you all can remember the legume family plants. Plants like beans, soya, and those stuff. So they actually have the root nodules. So there we consider a family of plants. So there we have the family. After that we go to almost the specific organisms, where we have the genus and finally species. So when you come to this level where you have the genus and species, you can directly point at a certain organisms. So for example, if you take humans, man, we say Homo sapiens. Homo is the genus, sapiens is the species. So using the genus and species, we identify each and every organism, the species of organisms. So that is the hierarchy of categories, and each one is known as a taxonomic level. So here, if we look at two examples given to you, that is again for extra knowledge, you can see here, consider the following examples to identify the taxonomic levels in a hierarchy of categories. So under that, two different examples are given. One is for modern human, Homo sapiens. When we say modern humans, we are actually the wise man. We are called the wise man because we can think. So we are the modern human. So that means there were humans before us also, but they couldn't think like that. They were not modernized like that. So there is a species like that as well. So if we look at the hierarchy or the taxonomical levels, if we take the domain, as I told you, we belong to Eukarya. This is domain Eukarya. That is one of the three domains, because our body is made up of cells with an organized nucleus. That is what is known as eukaryotic cell. We have discussed this term when we discussed the cell lesson as well. So, domain Eukarya, organisms that are made up of eukaryotic cells. Then we have kingdom Animalia. So, you all know what animals are. All the animals belong to this kingdom Animalia. Then we have phylum Chordata. What are chordates? In their early stage or the developed stage, now we have the vertebral column. But when we say chordates, they have a particular supportive rod-like, cord-like structure. So, based on that structure, they are classified as chordata. Then under chordata, there is the class that is Mammalia. Now, we have discussed many different classes. We belong to class Mammalia. Why? We nourish our young with milk, as well as the female organisms have a mammary gland. So, that is why class Mammalia. Then we come under the class Mammalia, we come in the order primates. Primates, all the apes, monkeys, lories, man, all of us come under the order primates. Primata, we are known as primates, order primates. Then under primates, we come under family Hominidae. the types of apes, chimpanzee, gorilla, ape and man, we all come under family Hominidae. So as you all can see, students, when we go down the order here, the number of organisms are being more selected and the types of organisms, the number is decreasing. So under family, it's a few types of organisms. Then we have the genus Homo, that is man. And sapiens is the species. So when we write the species, we write both genus and species together, Homo sapiens. Homo, we have similar characteristics and we are man. Wise man is the meaning of sapiens in Latin. So modern man comes under the species Homo sapiens. That is how we are classified in this natural classification. The same way, we can look at the classification for a coconut tree. So here, if we take the coconut tree or coconuts, you can see the name Cocos nucifera. Now we will go into detail about how we write this name, that is the scientific nomenclature, but for now we'll try to understand the taxonomical levels only. So again, domain Eukarya. So both animals and plants, I told you all, we are all made up of cells that have an organized nucleus. So eukaryotic cells. That's why we come under domain Eukarya. Same goes for the coconut tree. Then kingdom Plantae. You know the characteristics of plants. So kingdom Plantae. All the plants belong to kingdom Plantae. Then under that we have the division Magnoliophyta. Magnoliophyta means angiosperms or flowering plants. You are familiar with that term. Plants that produce flowers. So the division under kingdom, here we don't have the phylum, we have the division. That is something you need to understand. Under animals we had phylum, here we have division Magnoliophyta, that means angiosperms or flowering plants. Then under that we have a class Liliopsida. When we say Liliopsida, it refers to monocot plants. Liliopsida. So all the monocot plants, they are the class Liliopsida. Then we have the order Arachales. When we say Arachales, those are the palm type plants. Coconut, arecanut, palm tree, all of them are similar. Even if you look at the plants, you can see the similarity. So they all belong to order Arachales. Under that, if you go to the family Ericaceae, again coconut, arecanut come under Ericaceae, the coconut type palm trees. So that is the family. Thereafter you come to genus. Genus is Cocos and species is Nucifera. So again, that is the only species of coconut palm plant that is coming under the family Ericaceae. So Cocos nucifera, coconut tree. Now can you all understand these, students? So here of course, you don't have to worry about memorizing all these names. This is for your understanding. I explain the terms also so you get an idea how we group the organisms. Initially, at the higher level, when we look at the domain, we have a very large number of organisms there. But as we go down the order, from domain, kingdom, then to phylum or division, class, order, family, genus and species, you can see the number of organisms in each group becomes lesser and lesser. So we are approaching the organism more specifically with specific characteristics so that we can identify the organism by the genus and species name. That is what you need to understand here. So I am sure you all have got a good understanding of this hierarchical categories or hierarchy of taxonomical levels in natural classification. So with that, students, I will move on to the next slide. So here we look at the 3-domain system of classification before we look at the 3-domain system of classification, I want you all to look at the 2 main types of classifications. So the most appropriate system to classify organism is the natural classification. So far we have been discussing that, and you all know why it is most appropriate. Why? Because it shows the evolutionary relationships among different organisms, as well as it shows the relationships among the organisms of the same species. So because of that, different scientists have introduced different classification methods from past. So under natural classification, different people have attempted to classify organisms. So out of those, the most commonly accepted ones are these 2, the 5-kingdom classification and the 3-domain classification. But now, we currently use the 3-domain system of classification. So one of them is the 5-kingdom classification. So in this, the highest level is the kingdom. There is no domain. So there are 5 kingdoms, and this system was introduced by Robert Whittaker. Robert Whittaker introduced the 5-kingdom classification in 1969. That was also about 50 years back. And thereafter, Carl Woese, he introduced the 3-domain classification. That was in 1990. So the modern classification is the classification introduced by Carl Woese in 1990 with 3 domains. So you all have to remember, students, the 5-kingdom classification. So in the 5-kingdom classification, there is no domain level. We looked at the taxonomical levels. The highest one was the domain, but in this, there is no domain. It starts with the kingdom. So under that, the 5 kingdoms, kingdom Plantae, kingdom Animalia, kingdom Protista, kingdom Fungi, and kingdom Monera. So those are the 5 kingdoms. This is for your additional knowledge. I am giving you all the names of the kingdoms. You all don't have to go into detail of that classification method. But we will be discussing the 3-domain classification introduced by Carl Woese. So under the 3 domains, we looked at 1 domain. What was that? Domain Eukarya. Like that, there are 3 domains. The domain Archaea, domain Bacteria, and domain Eukarya. So I am sure you all can understand that concept. With that, students, I will move on to the next slide. The 3 domains are. So if we write the 3 domains, we will have to always write it as, or better to write it as, domain Archaea, domain Bacteria, and domain Eukarya. So students, you all can see here domain Archaea, domain Bacteria, and domain Eukarya. So as I told you all, we will look at this diagram. Now here you can see from the common ancestor, we have the 3-domain classification. Why do we say common ancestor? You all can remember during the evolutionary process, there was the pre-cell, and from there it was the bacteria, anaerobic heterotrophic bacteria, and thereafter the different organisms develop. But this evolutionary process, the development of different species, took different paths. So in that, if we look at the common ancestor, from the common ancestor, here you can see there is one branch, there is another branch, and this branch leads to the second domain, domain Bacteria. So here we will have domain Bacteria. So that developed as one separate branch. Whereas the other separate branch, it led to the division of 2 different domains. That is domain Archaea and domain Eukarya. Both of them came from this particular branch. So here we have Domain Archaea, and here we have Domain Archaea. So from this diagram, students, you all can understand bacteria developed from the start, from the common ancestor as an evolutionary path. On the other path, or on the other side, the branch, you can see up to this point, the development has been common. Thereafter, there is a division, and that develops into Domain Archaea, and on this side, there is a development up to Domain Archaea. And all the organisms that we have on Earth now can be classified into these three domains. And out of these three domains, Domain Archaea has the most advanced animals and plants as well as all the organisms, and it has a very large number of species there. So that is the introduction to three-domain classification. And under this, we will be discussing all the three domains and the kingdoms coming under Domain Archaea as well. So Domain Bacteria and Domain Archaea, we will discuss the common characteristics and examples of organisms, and we will go into detail about Domain Archaea. With that understanding, students, I will move on to the next slide. Domain Archaea. So when we say Domain Archaea, we have certain organisms. I told you all these are organisms that have a prokaryotic cell. So therefore, we call them prokaryotes. They are known as prokaryotes. So cells that do not have a definite nucleus or does not have an organized nucleus. So those are known as prokaryotes. So the organisms that belong to Domain Archaea, they are all prokaryotes. So if you look at the nucleus structure, they do not have an organized nucleus. So here we call it the prokaryotic cell. Prokaryotic cells, because they do not have an organized nucleus. I'm sure you all can understand that. So if we look at the environment in which Domain Archaea organisms live, they can live in extreme environmental conditions. We have discussed this before also. Extreme environmental conditions. What's the other name given to extreme environment? Hostile environments. So here we can also say hostile environments. So when we say extreme or hostile environments, volcanoes, deserts, hot springs, ocean beds, and high saline environments and polar ice caps. So what is the extreme condition there? Volcano, very high temperature, lava. Then deserts, very, very dry, no water or very, very low amount of water. Hot springs, again very high temperature. Ocean beds, the depth is greater, sometimes pressure is high, the salinity is high. So then again, high saline environment. Environments with very high salt concentration. So those are high saline environments. And again, polar ice caps, the temperature is very, very low. So these are all extreme environmental conditions or hostile environmental conditions. So we normally can't survive in these conditions. But the prokaryotes that belong to Domain Archaea, they are called methanogens and halophiles, two types, methanogens and halophiles. They can live in all these extreme environments. Then if we consider another characteristic, sensitivity to antibiotics. Now you all know what antibiotics are. Antibiotics are chemical substances produced by one microorganism to sabotage or destroy another microorganism. So for example, penicillin is an antibiotic obtained from the fungus Penicillium. And this is used against the function of bacteria that you all know. But here, as I told you all, Domain Archaea includes organisms that are known as halophiles and methanogens. They are not affected by antibiotics, or we can say they are not sensitive to most antibiotics. They are not sensitive to most antibiotics. That means you cannot destroy or sabotage the Domain Archaea organisms by using antibiotics. That is the meaning. Then if we look at examples, as I told you all, those are the methanogens, and halophiles. Those are the two types of organisms that belong here. When we say halophiles, normally they are salt-loving, or they like to live in extreme salt conditions. So those are the halophiles. Then we have the methanogens. They have cells without an organized nucleus, so they are made up of prokaryotic cells, and they can live in extreme environment conditions. So to understand how these organisms look like, now I will show you all a video where you can have a look at the examples of these two types of organisms. Organisms that belong to Domain Archaea. Okay, students. So you all were able to see the structure of these archaea organisms. So as I told you, two types of organisms, methanogens and halophiles. The names actually refer to their characteristics. So as I told you, halophiles, they are salt-loving. So they prefer to grow in saline environments. But at the same time, if you look at the methanogens, now they actually produce methane gas as a byproduct of their metabolic activity. So that is why we call them as methanogens. They evolve methane gas. So from that you all can have an idea as to what these organisms are. So this is all that you need to know for Domain Archaea. What they are, the prokaryotic cell organization where they can live and their sensitivity to antibiotics, and also the examples. So with that, students, I will move on to the next domain, that is domain bacteria. So domain bacteria. So under domain bacteria, there are two main types of organisms, bacteria as well as cyanobacteria. So when we say cyanobacteria, they also have the term bacteria in their name. So you can easily remember both those types of organisms belong to domain bacteria. So first I will write down the examples. This is bacteria and cyanobacteria. We give another name to this cyanobacteria. They are also known as blue-greens, or we call them blue-green algae. You're familiar with bacteria. We have discussed bacteria and uses of bacteria, the harmful effects, so you're familiar with that. But the cyanobacteria, they actually resemble algae. You all can remember the algae cladophora. That is like a ribbon-like structure, and they have the chlorophyll pigment, so they do photosynthesis. In a similar manner, the cyanobacteria, the blue-greens, resemble algae. They also have green color pigment, chlorophyll, so they can do photosynthesis. So those are the two main types of organisms that belong to domain bacteria. And if we look at their features. Characteristics of domain bacteria. Again, if you look at the organisms, both bacteria and cyanobacteria, you all can remember, students, bacteria is microscopic and they are unicellular. They only have one cell. Although these cells combine and form like a long chain-like structure, still they are made up of one cell. So they are microscopic and unicellular, and also they are prokaryotic organism. Now you all know the meaning of prokaryotic. What is it? They do not have an organized nucleus, or they do not have a defined nucleus. So that is why we call them prokaryotic organisms. So the organisms are microscopic, unicellular and prokaryotic. Both bacteria and cyanobacteria, they are the same. So again, cyanobacteria also, they are unicellular, so one cell, but these cells can combine and form like a long strip-like structure or a ribbon-like structure. We will look at the pictures after this, then you will know. So here they do not have an organized nucleus because they are prokaryotes, or because they don't have a organized nucleus in the cell, they are prokaryotic cells. Then if we consider the environment, they can basically live in any environment. You can remember when we discussed microorganisms in the previous grade, they live in all environments: soil, water, atmosphere, then within and outside the living organisms, bodies of living organisms, man, plants, animals, in all living organisms. Then they can live in dead plant and animals as well. They act as decomposers, you can remember that. And also they can live in hostile or extreme environments. So they basically can be found everywhere in the environment, everywhere, no exception. So you can get bacteria and cyanobacteria basically everywhere on earth, wherever there are living organisms. Then if you consider the sensitivity to antibiotics, I told you all penicillium. Penicillium is a fungus that produces, or from that fungus we get the antibiotic penicillin, and we use that against the bacterial infection. So that means bacteria, they are sensitive to antibiotics. So this is a difference. So now can you all understand, students? Both domain archaea and domain bacteria, they are both prokaryotic cells. They do not have an organized nucleus. But when it comes to sensitivity to antibiotics, archaea, they are not sensitive. But bacteria, they are sensitive. So there are similarities and differences between these organisms. So that is why this particular classification is very, very useful. You can understand both the characteristics, similarities as well as differences. So bacteria, they are sensitive to antibiotics. So that means you can control the growth of bacteria using antibiotics. So we can treat diseases caused by bacteria using antibiotics. Then if we look at the other features. Now some of these are pathogenic. What is the meaning of pathogenic? They cause diseases. So they cause diseases. You all can remember different diseases caused by bacteria. So they live in the host cell and they cause diseases to the host. The host can be man, plants or animal. So those are the diseases. But some of them, here it says some are pathogenic. Why is that? Because bacteria is pathogenic when they cause diseases. But as I told you, cyanobacteria, they have chlorophyll. Then they can produce their own food. So they don't become pathogenic. So you have to remember that. And also under bacteria, you can remember, students, there are nitrogen fixing bacteria. There are other bacteria that are used for the production of curd, yogurt, like lactobacillus, streptococcus. Then we have rhizobium, azotobacter. They don't cause diseases. So they are not pathogenic. And the decomposers, they are also not pathogenic. But the bacteria that cause diseases, they are the pathogenic bacteria. You all have to remember that. That is why we say some are pathogenic. And then if you look at the other information or other features, they are the most abundant group of organisms. So most abundant, they are spread throughout the environment, throughout the biosphere, everywhere in the environment, and you can find them abundantly. The number is high. So that is a feature of domain bacteria. So these are the characteristics that you all have to remember, students. We will be comparing these groups again also, but for now, understand these features. We will compare the different groups, bacteria, then algae and fungi as well. So before that, I will move on to the next slide. Here you all can see the diagrams. Bacteria, you can see this shape. This is a rod or rectangle-like shape, and that is known as a bacillus bacteria. Bacteria have different body shapes, as I told you. When we go to that particular table, we will discuss all these in more detail. But here you can see this particular shape. So these are again prokaryotic cells, unicellular organisms, all are just single cells. And it has this particular shape, bacillus. Similar to that, it can have a spherical shape. Then we will be calling it coccus. So different shapes. Then here you can see cyanobacteria. That again you can see the unicellular bacteria. But as I told you all, you can see this tape-like structure. So there also you can see this greenish color nature. Cyanobacteria. So they can live as a community as well. They can spread throughout in environments. So these are the two main groups or two main examples that come under domain bacteria. Now we will watch a video to understand about these bacteria and cyanobacteria in more detail. So when you watch the video, you can identify and understand the shape of different types of bacteria and cyanobacteria. Okay, students. So from the video, you would have got a better understanding about what these organisms are. Bacteria and cyanobacteria or blue-green algae that belong to domain bacteria. So in that diagram, you all can remember with the common origin, from the common origin, there were two branches. One branch led to the domain bacteria. The other branch goes and divides again to form Domain Archaea and Domain Eukarya. But out of the three domains, both Domain Bacteria and Domain Archaea, they have prokaryotic cellular organization. So under that, we have discussed the two types of domains, and you all are familiar with the bacteria and cyanobacteria. So with that, I will move on to the next slide where I discuss a typical cell of bacteria. So this is again given to you all for extra knowledge. This is an electron microscopic structure of a bacterial cell. So here this is the bacterial cell. This is usually a typical cell. You all know the meaning of typical cell. What is a typical cell? A model of a cell that includes all the possible structures that can be present. And here, electron microscope structure. So here students, electron, there is no E, it should be ELECTRON. Electron microscopic structure of a bacterial cell. So if you look at this bacterial cell, again here you can see the shape. I explained this shape is called a bacillus shape. So the cell is in this shape, like a rod-like or oval shape kind of. So that is a bacillus shaped cell. So in that, if you look at the parts of the cell, here you can see ribosome. You are familiar with ribosomes. When we discuss the plant and animal cells, we came across ribosomes. In ribosomes only protein synthesis takes place. And also you can remember ribosome does not have membranes. It has two bodies, a small body and a large body. That is the structure of ribosome. So same as that, we have ribosomes, you can see scattered all around the place, and this is the cytoplasm. You can see the cytoplasm. In other cells also there is cytoplasm, a gel-like structure in which all the other organelles are embedded in the cytoplasm, or the other organelles or the structures of a cell, they are present in the cytoplasm. So here also this is the gel-like structure, the off-white color area, that is the cytoplasm. In that you can see these dots, those are the ribosomes. So the ribosomes, they actually do protein synthesis. Then you can see nucleic material and you can see it is DNA. So here this fiber-like structure or thread-like structure, that is the nucleic material DNA. So when we discuss nucleic acid, you all can remember students, normally DNA stores genetic information and carries genetic information from one generation to the other in most of the organisms. But in some bacteria, we said there can be either DNA or RNA. So you all have to remember that. So here also there is DNA shown to you, but that is the nucleic material. The same thing, here you can see this particular structure that is called the plasmid. Now plasmid is also a DNA, usually a circular shaped DNA. So that is also DNA. A circular DNA is known as plasmid. So that is also present in a bacterial cell. Then you can see around the cytoplasm there is the cell membrane. Now this cell membrane is a lipoprotein membrane, like the cell membrane present in other cells. Like in plant cells, animal cells, all of them have cell membranes. A similar cell membrane is here also. So bacterial cell membrane is same as the cell membrane of the other cells, the eukaryotic cells. Although this is a prokaryotic cell, the cell membrane is the same. So you have to remember that. That again acts as a semi-permeable membrane or selective permeable membrane. You can remember that as well, students. It doesn't allow all the substance to go in or out of the cell. It selectively allows substance to go in and out of the cell. So that is the cell membrane. Around the cell membrane, you can see the cell wall. So bacteria also have a cell wall, similar to plant cells. You all can relate the properties, students. Some are similar, some are different. So you have to learn it that way. When you understand, you need to compare as well as you need to identify the similarities and differences. So cell wall is there. As I told you, it will allow substance to come in or go out. So it's like a boundary. So it's there for protection, cell wall. And thereafter, there is an envelope. Now that is also there as a protection. Envelope is usually a multi-layered structure. Around the cell wall, there is the envelope. So that is a multi-layered structure that is there for protection. Then we have two more structures. There you can see flagella. Where else have you all come across flagella? Chlamydomonas, an algae that has flagella. Then Euglena, that is a protozoan that also has flagella. What is the function? It's there for the locomotion of the organism. So for the movement of the organism. So here flagella is there for movement. So the bacterial cell uses flagella to move around for their movement. Then we have this pili. Now you can see the difference, students. Flagella is a long, thin, thread-like or hair-like structure. You can imagine it that way. Now, this is an electron microscopic view. So, it's very, very tiny, but still you can imagine it like a long, thin, thread-like structure. Whereas the pili, they are very short, thin, thread-like structures. Now, the function of this pili is to attach the cell. Attach the cell to surfaces. Now, we said no, these bacteria can live on any environment. It can live on our skin, it can go into our body. So, when it is there on our skin, it uses this pili to attach itself to the skin. So, any surface, the pili is used to attach the bacterial cell to the surface. So, although this is for your extra knowledge students, you all have to have an understanding or idea about the structure of this cell. So, with that students, I will move on to the next slide. So, here, now that we have understood the characteristics of bacteria, we will look at several harmful and useful effects of bacteria to human are given below. So, here we are going to look at harmful and useful effects. So, under that first we will look at the harmful effects of bacteria with examples. So, first if we look at the harmful effects, the first one you all know, they cause disease to man, plants and animals. So, under this, we are mainly looking at the effects caused to humans. So, you all know bacteria causes disease to plants and animals as well. But since we are discussing only the effect to humans, I will only write down the diseases caused to man only. So, as examples, we can consider the disease tuberculosis. Tuberculosis is a disease related to the respiratory system. It's caused by a bacteria. So, when these bacteria cause diseases, what do we call them? They are pathogenic bacteria. You have to remember that term as well. They are pathogenic bacteria. So, we said when we were discussing the characteristics, some are pathogenic. So, those are the bacteria that are going to cause these diseases. We have tuberculosis, then we have pneumonia. Pneumonia again is a disease related to the respiratory tract. You get phlegm, you get high fever, and that might lead to complications as well. Pneumonia. Then we have leprosy. Leprosy, a disease to the, that is related to the skin as well as the nervous system. It can also affect your eyesight. So, that is a somewhat complicated disease caused by bacteria, leprosy. Then there is diarrhea. Diarrhea is again a disease caused to the digestive system, involved with the digestive system caused by bacteria. Then there is tetanus. Tetanus is also known as lockjaw because when there is tetanus, the muscles become stiff. You will be unable to move your jaws or your muscles. So, because of that, we call it lockjaw. So, these are diseases caused by bacteria: tuberculosis, pneumonia, leprosy, diarrhea and tetanus. So, that is one harmful effect. They act as pathogens and they cause diseases to man, and also they cause diseases to plants and animals. But as I told you, since we are looking at the effects of bacteria to human, we only stick to the diseases caused to humans. Then the second effect, harmful effect. What is it? Food spoilage. You all know most of the microorganisms, especially bacteria, fungus, they all cause food spoilage. Food becomes unsuitable for consumption. They change the nature of the food. The color changes, odor changes, then the nutritional value changes, appearance changes. So finally, it becomes not suitable for consumption. So that is also a harmful effect. Food spoilage caused by bacteria. That is the next one. Food spoilage. Food spoilage caused by bacteria. So two main harmful effects, causes diseases and food spoilage. You all have to remember these, students. Then under food spoilage, we have discussed in grade 9 the different types of food spoilage based on the substance that is present in food and all that. So you're familiar with all these, but here as an example for the harmful effects of bacterium, you need to understand causing diseases and food spoilage. With that, I will move on to the next slide where we discuss the useful effects of bacteria. So to produce curd, yogurt and cheese, that is one useful effect. What are the bacteria? Can you all remember the names? Curd, yogurt and cheese. Lactobacillus, streptococcus. So lactobacillus, now here students, you all know these are scientific names. When it is printed, it's printed in italics. You will learn the rules towards the end of this lesson. When we are writing the name, we will have to underline it. So lactobacillus and streptococcus. They are both involved in production of curd, yogurt and cheese. Lactobacillus, streptococcus. Then the next use separation of fibers from coconut husk, agave leaves and also to tan leather. So there are two uses. This is one, separation of fibers from coconut husk, agave leaves. You know these examples. What do we use there? Normally, it is a bacteria that is used to dissolve the pectate. You can remember that, students. Pectate is a paste-like substance present between the fibers of these plants, coconut husk and agave leaves. So when you use the bacteria, you soak this fiber in water, there is bacteria that has an enzyme called the pectinase enzyme. So this pectinase enzyme is used to digest pectate. So these are all things, students, that you have already learned. That's why I'm just giving you all the points to recall your memory. So pectinase enzyme is produced by the bacteria. Then there is the next function, that is to tan leather. What is tanning of leather? Preparing the leather for, like you get the skin of animals and you process it to produce the leather, so that can be used to make leather goods. So for that process also we use bacteria, tanning of leather. Then to fix atmospheric nitrogen to increase soil nitrate level. You're familiar with this process as well. There is about 78% of nitrogen in the atmosphere, but that cannot be absorbed by the plant. So that nitrogen has to be converted to a form that can be absorbed by the plants. So that is done by bacteria. Some are symbiotic bacteria. Earlier also I mentioned in the legume plants. There are the root nodules. In that root nodules, there is a bacteria that lives symbiotically. What is it? Rhizobium bacteria. So you can remember the name, students. Rhizobium bacteria. Again, students, this is a scientific name, so I'll be underlining it. If it's printed, you can see look at your textbook. It is in italics. Here it is underlined. Rhizobium bacteria. Then there is the free-living bacteria that also converts atmospheric nitrogen to forms that can be absorbed by the plant. What is that? Azotobacter. You can remember that as well. Azotobacter. Azotobacter is different from Acetobacter. So you all have to remember that, students. Azotobacter. That is a free living bacteria. Here also we have to underline. Azotobacter, it is a free living bacteria. Rhizobium is a symbiotic. It lives in the root nodules of legume plants. They both fix atmospheric nitrogen to increase soil nitrate level. So to increase soil nitrate level. That is a useful effect. Then finally, to decompose dead bodies and structures. Decomposing dead bodies and structures, those are the decomposers. We call them decomposers. So bacteria act as decomposers. So they decompose dead bodies and structures. Even methanococcus, in the production of biogas, methanococcus is used, and that also decomposes organic material. So that also can be considered as an example, methanococcus. So if I write that here, methanococcus. In addition to methanococcus, there are many other bacteria that acts as decomposers. So here they are decomposers. So they live on bodies of dead plants and animals, and they decompose that material. When we say decompose, they convert the complex organic substances to simple forms. So that is again a useful effect of bacteria. So if I again go back, we were trying to understand the useful effects of bacteria to man. Under that, producing curd, yogurt and cheese, then separation of fibers, tanning of leather, then fixing nitrogen, and as well as the decomposing process. So all these are very useful in many ways. Now students, why did we discuss these useful and harmful effects? When I introduced classification, you all can remember under significance of classification, we said that by classifying organisms, we can identify organisms that are economically useful to us. So here you can see some of the economic uses. The same way I told you all the harmful effects. They affect us in many ways. If you fall sick, if the microorganism affects the plant crop, then there is an economical effect. It can be harmful as well. So both are important to us. So we can understand these when we classify organisms more easily. So with that students, I will move on to the next slide. This is again for extra knowledge. This is an organism or some living organism that has few living characteristics, or basically 2 living characteristics. Virus. You're familiar with virus. So here, virus was first observed by a Russian scientist named D.J. Ivanovsky. Students, normally when we spell D.J. Ivanovsky, he is actually Dmitry Ivanovsky or Ivanovsky. He is a Russian scientist, student, so don't worry about the pronunciation of the name, but this is how normally it is spelled. So there is a difference in the spelling, but just remember about the scientist Dmitry Ivanovsky. So he is the scientist who identified viruses. That is in 1892. So they are not identified as living organisms. Why? You all know, because they don't show many of the living characteristics. They don't have a mode of nutrition, or they don't produce their food, or like they are not autotrophic or heterotrophic or anything. At the same time, they don't respire, they don't have metabolic activities, they don't have a cellular organization. So most of the living characteristics are not there. They show non-living characteristics because they exist as crystalline structures outside of host cells or living cells. But inside the living cell, they become active, they multiply inside the living cell, and they cause diseases to the host. So they possess both living and non-living features. The only living feature of them is the multiplication within the host cell. While they multiply, they also cause diseases. They cause diseases. So here there are some examples given to you, students. You can see electron microscopic view. Again here it has to be electron microscopic view. You know electron, E-L-E-C-T-R-O-N. Electron microscopic view of several viruses are given to you. You can look at the first one, AIDS virus. That is also known as HIV virus. Human immunodeficiency virus. So that is the virus that causes the disease AIDS. You can look at the structure of that. Then there is the bacteriophage. Again, bacteriophage has to be spelled without the R there. B-A-C-T-E-R-I-O-F-A-G-E. Bacteriophage virus. From the name, you all can relate it to something, students. These viruses actually infect the bacterial cell. That's why we call them bacteriophage viruses. So this is the structure of a bacteriophage virus. Then we have the bird flu virus. Again, as the name implies, they cause a flu in birds, common among birds. So when you all say flu, normally it's related to the respiratory system with fever and all that. But we also can be affected by this bird flu virus. So as you can see, all the viruses shown here, they cause diseases. In addition to that, you all know about the common flu virus, the common cold virus. So that also is a virus that causes the disease common cold. So all the viruses, they cause diseases and they show the living characteristic of multiplication within a host cell. Otherwise, they show non-living characteristics. That is the reason why these viruses are not classified under the 3-domain or 5-kingdom classification, because only characteristic is that they multiply inside the cell and cause diseases. So other than that, they are non-living characteristics. So therefore, we do not have them under the scientific classification or the natural classification. But as we have discussed before, you all know, because they are electron microscopic, they are not visible to our naked eye, scientists put them under microorganisms. So we study them under microorganisms, but they are not part of the classification. That is something that you all have to understand, students. So with that, I will end this chapter. So in this chapter, students, we discuss the natural classification. Under natural classification, the 2 main types, the 5-kingdom classification by Robert Whittaker. You all just need to know that classification is there. You don't have to go into detail, but we discussed the 3-domain classification, and under that, I have discussed the first 2 domains, Domain Archaea and Domain Bacteria. So in the next chapter, I will discuss the Domain Eukarya and the groups under that.